Looks like people are joining the webinar now. We've got a few people with us. So welcome everyone to our Fall Grant Program update. I'm Susan Hedstrom, Executive Director of FPWR, and I'll be your moderator today. We're happy to have you joining us and would like to answer any questions you may have. So to ask a question, simply type your question into Zoom's question text box, and we'll answer as many of those questions as possible at the end of the presentation. FPWR plays the important role of matchmaker and collaborator by bringing together experts from around the world to tackle the hard challenges of PWS. Since 2003, we have invested over $16 million in high-impact grants. We've advanced our understanding of PWS, funded PWS clinical trials, and have developed the critical tools needed to support PWS research. FPWR funds 2 grant cycles each year, and today, Dr. Theresa Strong will provide highlights from our most recent grant cycle. Most of you likely already know Theresa, but for those of you who may be newer to our community, Theresa is founder of FPWR, our Director of Research Programs, and the mother to a young adult with PWS. Theresa, we're ready for you to begin.
Great. Thank you so much, Susan. I'm excited to talk today about some of the new projects that we'll be funding in this grant cycle. And also excited to be participating, or through my son anyway, in the Harvesting Hope Turkey Trot. We're excited that both the boys of my life, my husband and my son, are both participating, and Daniel has run or walked more than 80 miles in the last probably 5 or 6 weeks. So it's been great for him. Today we're going to talk about just a very brief update on research developments in PWS. And then I'm going to spend most of the time talking about the new grants that we've recently funded. And as Susan mentioned, we have 2 grant cycles per year. These are the grants that were submitted last spring and that are being funded this year. And then we should have time for questions and discussions. I welcome your questions. As Susan said, please type that into the Q&A box. And I've got FPWR's mission down here. Always a good reminder of exactly what we're focused on. And we are always focused on advancing research so that we can get new therapeutics to our kids and adults with PWS. So I want to start with the bad news and kind of get that out of the way, which is, as many of you know, we recently had an FDA advisory committee meeting focused on PWS. There was a great deal of excitement in the community because this is the first time a new drug application has gotten to this point in the drug approval process in more than 20 years. So Levo's drug LV-101 has completed a phase 3 clinical trial that showed some positive results. Unfortunately, the advisory committee, which is an independent group that the FDA brings together to provide guidance to the FDA, the advisory committee voted 12-1 that the data gathered so far had not met FDA's standard of substantial evidence of effectiveness for treating PWS. So the FDA usually takes the guidance of the advisory committee. So we expect that the FDA will agree that there needs to be more work done to show that LV-101 is effective in PWS. So this was obviously very disappointing for our community, as we had hoped that this drug would be approved at this stage. But we do look forward to working with Levo and seeing what needs to be done and then getting that done. It is also notable, and I think a small silver lining, that the FDA and the advisory committee both really seem to understand the challenges of PWS. They appreciate the unmet need in our community. They are really supportive of the general idea of developing therapeutics for PWS. And so I think if we can, reach that goal of substantial evidence of effectiveness in their eyes, that we can get some new therapeutics approved in PWS. If you're interested in more details, I have the link down there for all of the materials from that meeting. And just want to acknowledge our work in collaboration with the members of the PWS Clinical Trial Consortium, as well as PWSA USA, in, again, helping the FDA understand the issues in our community and, be supportive of the need for new therapeutics for our kids and adults with PWS. And I did want to mention that, we have a very active pipeline of therapeutics that are in clinical trials for PWS. I mentioned Lebo, which has completed a phase 3 and unfortunately, has not yet met that substantial evidence of effectiveness. And so presumably they will have to do additional work in the PWS space to gain that approval. Soleno Therapeutics has also completed a phase 3 study. They are also talking with the FDA and have submitted additional confirmatory evidence. They believe in support of the use of DCCR diazoxide in individuals with PWS, and we expect that the FDA will have some communication back with them sometime in the coming weeks. There are also many clinical trials that are open, both sponsored by industry as well as in academic settings on a number of compounds, including oxytocin, pitolisant, which is a wake-promoting drug in PWS. Oh, I got that wrong. Sorry. Tesomet, which is Saniona. Oh, sorry, that was wrong. Tesomet, which is from Saniona, which is a drug aimed at reducing hyperphagia. And then Radius has a CBD-based synthetic CBD, looking at both hyperphagia and anxiety in PWS. And then some newer companies, Aardvark Therapeutics, for example, has a new drug that will be testing in a small clinical trial. So there is a lot of activity in the PWS field, which we are very fortunate to have. What we need now is to make sure that these clinical trials get done in a reasonable amount of time. And so we would urge you to be aware of the clinical trials that are ongoing and think about enrolling in those clinical trials. And you can find more information on FPWR's clinical trials site. We have information about the individual clinical trials that are ongoing, also provides information about the inclusion and exclusion criteria. So you can see if your loved one with PWS is potentially eligible to participate. And it also provides the sites that are available. For most of the industry-sponsored trials and for some of the academic trials as well, even if you need to travel, there is often support so that you don't have to pay for travel to the clinical trial site. In addition, a lot of the companies have really learned some great lessons from COVID and really have worked to make the trials much more accessible to people who are far away from a clinical trial site. So having remote visits, for example, or having a home health nurse come to your house to ease the burden of actually participating in a trial. So even if you've looked at trials before, I would urge you to take a look now and see if it might be something that you can manage in your life. I also wanted to mention the conference updates. We had a great virtual conference last month, and we've gone ahead and put all of the different sessions on FPWR's YouTube channel, which is listed here. So if you want more information about some of the clinical trials that are opening or are currently open or about to open, you can go ahead and watch the clinical trial session. We also have a research roundup that talks about a lot of the research that was presented at our scientific conference. A lot of that is FPWR supported work if you're interested in learning how those funds are being used. And we also have the session, What's Happening in PWS Research, which is really focused on the things that the FPWR team is doing to advance and support PWS research. There's a lot of other things, with sessions on behavior and anxiety and sleep and exercise. So I would just urge you to go to the FPWR channel and maybe have that playing in the background as you eat your lunch or something. I also wanted to mention one of the activities that we're really excited about is the development of the PWS Clinical Investigation Collaborative. And this is a collaboration of about 35 clinical sites across the US and Canada. Many clinicians who are familiar to you are participating in this: geneticists, endocrinologists, GI doctors, orthopedists, all coming together to help advance the care of individuals with PWS. So the mission and vision are shown here. I won't read the whole vision because there's a lot going on there, but the mission is to improve the quality of clinical research and medical care for people with PWS across the lifespan. And the members of the CLIC are doing this through a collaborative investigation and research to support evidence-based care. So one of the activities that they're doing is figuring out how to share information about caring for individuals with PWS so that A, they can learn new things about the complications that our kids experience, and B, they can develop new standards so that across the country and in Canada as well, everybody has better access to excellent care. I also wanted to mention the Global PWS Registry, which, as you know, is a very active registry to gain information about the complications that our kids experience. The members of the CLIC are using the registry to better understand the patient perspective on those complications. We're also using the registry to advance our understanding of things as diverse as, the incidence of strabismus or lazy eye in PWS, and the behavioral issues that some of our older kids experience, including, what is the rate of suicidal thoughts and behavior. We have a paper that just came out on that, which showed that the rates of those kinds of thoughts are not really very different than the general population, but they have different characteristics. They tend to come on later. The split between males and females is different than in the general population. And that kind of information will be helpful, I think, as for both families and clinicians to be on alert and be able to recognize and manage those behaviors. Please look in the next few weeks. The members of the PWS CLIC are really interested in understanding about feeding tubes and complications of feeding tubes in our population. This is an opportunity for those of us who have older kids to kind of look back and see, whether there were any long-term complications in our kids. And it's an opportunity for parents of younger kids to provide information about sort of what is currently being done in children with PWS, what kinds of feeding tubes are being used, and for how long. So those kinds of questions. So it's an opportunity for all of us to pool our data and really help these clinicians, develop a best practices around using feeding tubes in infants with PWS. Okay, so now I'll move to our grant program, which, this is the spectrum going from research and discovery across the development of new medical products, and then the implementation of that in the care of individuals with PWS. FPWR has a lot of programs that are funded to support this pipeline all the way across. But what we're talking about today is our grant program. And our grant program funds projects anywhere along this pathway. And these are what we call investigator initiated, in that scientists, come up with an idea, an area where they think there is opportunity to gain understanding in PWS, and they submit their projects to us. And we, of course, review those with expert scientists as well as parents, so that we select those grants that are both scientifically sound and relevant to our community. So this program is the program that we started at the inception of FPWR and has been going on since then. And so we see a lot of returns on those investments. And again, they're really diverse things that have come out in the literature that have been supported through this program. This slide just shows you some of the recent papers, everything from molecular changes in PWS neurons, looking at why some of our kids have more autism features than others. And that is work by Dr. Ryder, through using mouse models to understand how oxytocin treatment might change the brain of individuals with PWS during therapy. Through developing new endpoints and looking at the cost of taking care of someone with PWS and other imprinting disorders. And that's really important for laying the framework, both for, paying for new drugs for PWS, as well as newborn screening for PWS. So understanding those costs, the economic consequences of PWS is really important for that. So I would just urge you, we try to keep our website up to date with the projects and then the outcomes from each of those projects, so you can see how the funding is being used to advance the science. And you can look at all of these sources on our website for that. So here's the list of the grants that have been funded this time. You may remember, if you've been coming to our webinars often, that in the spring we only funded, I believe, 5 projects. That was because we were coming out of COVID. The finances weren't, quite clear. Hopefully we're getting back on a very good footing. And so for that reason, and also because we had a lot of really good proposals in this last round, we have funded more in the second half of 2021. So I will run you through most of these. I'll just start by saying 3 of these were a second year of funding. So these were projects that we funded previously, some of them in 2019, some of them in 2020, for example, that have now competed for a second year of support. So I won't go into much detail about this since we have so many others, except to say one of them is in the development of a gene therapy for obesity in PWS, and that is a study at Ohio State University. And it's really looking at a particular pathway, the BDNF pathway, which is a brain-derived neurotrophic factor, so a sort of a hormone in the brain that helps neurons grow, and seeing if tweaking that system can improve the metabolism in PWS. The other 2 are really very basic science. We still do not have a complete understanding of the function of all of the RNAs from the PWS region. And that's really important if we're going to develop targets for therapy that develop them in a very rational way. So this program is our program by which we try to advance that science. And 2 of the projects will look at different aspects of the RNA and the genes in the area, looking at their basic function. So of the new projects, so these are newly funded, not second year studies, there are 5 that kind of fall into this group that I called model systems of PWS for target discovery and therapeutic development. And again, a lot of these studies are looking at the basic biology of PWS, looking for how PWS is different than the typical development of neurons or mice in some cases, and using that information to identify new drugs and new pathways that might be targeted by drugs. So I'll go through each of these. The first one is the development of a new mouse model for PWS. And this is from Dr. Muscatelli and Dr. Boret, who are in France and who have worked collaboratively for a number of years. Dr. Muscatelli has expertise in PWS mouse models. She's done a lot of the work looking at, or that initial work that showed that oxytocin in the newborn period can correct a lot of deficiencies in PWS in a mouse model. And she has worked with Dr. Boret, who is a neurobiologist who looks at circuitry in the brain. So it's a really nice collaboration. And they've done a lot of really interesting work to understand how the brain development is different in PWS compared to typical, well, in this case, mice. An issue with the PWS mice is if we knock out that entire region as it is in humans, the mice typically don't live. And so the field has taken to deleting one gene at a time. And that is valuable and tells you about each gene, but it leaves something to be desired because some of these genes are related. And so when you knock out one and leave the other gene can compensate for the one that's been knocked out. So in this case, Dr. Muscatelli developed a mouse model in which nectin and MAGEL2, which are 2 related genes that sit right next to each other in the critical region, are deleted at the same time. And they're going to be characterizing the behavioral, developmental, and metabolic characteristics of these animals. So they've newly developed this animal model and now will be doing all of the characterization. So we're excited about this study because this is a team that we know works really well together. They get things done. They understand PWS. And so there's a high likelihood that they'll gain new information from this double knockout. And the other thing is that, all of our mouse models with PWS, they provide us good information, but there are some limitations. And the thought is with this mouse model that is knocking out 2 related genes at the same time, it may be a more relevant model for testing new therapeutics. So the new mouse model is expected to be more relevant to understanding how the brain develops and how the behaviors are different in PWS, and also, again, provide a model for testing new therapeutics. The second study is looking at the role of the oxytocin receptor expressed in astrocytes in PWS and Schaff-Yang syndrome. So this is a study by Dr. Schaff of Schaff-Yang syndrome. Has recently been a discovery that a certain support cell, so these astrocytes they're very numerous in the brain and they provide support to neurons. And it actually turns out that they have oxytocin receptors and that they are integrated into that responsiveness to oxytocin. So Dr. Schaff is going to be examining the structure and function of these astrocytes in brain regions in a PWS mouse model to see how they're different in PWS compared to typical mice. We're excited about this study because this is an experienced PWS researcher and Schaff-Yang syndrome researcher, so he knows the field well. He understands the deficits in oxytocin in individuals with PWS and Schaff-Yang syndrome. We know that the oxytocin circuitry is disrupted in PWS and in these mouse models, and this will really look at how and why that circuitry is disrupted and may be very useful in understanding how we should target therapeutics. So we expect that this project is going to provide insights into how changes in the astrocyte biology and oxytocin response might underlie some of the cognitive, particularly the cognitive and the social aspects of PWS and Schaff-Yang syndromes. And again, maybe guide us in how to develop targeted therapeutics to address that. We next have Dr. Larry Reiter, who is going to be looking at delayed neural development in PWS neurons. So Dr. Reiter's group, for those of you who have been following FPWR for a while, his group has been studying neurons that come from PWS baby teeth. So many of you have sent your child's baby teeth to the Reiter lab, and they use that to grow out neurons. And it gives them a great source of neurons from lots of different patients. So they're able to look at the molecular characteristics. And they look at PWS neurons, but they also look at, typical neurons and then other neurodevelopmental disorders. So they have a really nice bank of neurons to look at. So what they found, and what we've heard a little bit from other researchers, is that the PWS neurons seem to have a different timeline of maturing than typical neurons. And this may be indicative of an underlying issue in PWS that is related to some of the social and cognitive and developmental issues that we see. So with this study, they're really going to focus in on that, looking at a larger sample set across more patients. And the team is also going to be taking genes and pharmacological agents and seeing if that can restore a more normal maturation process in these PWS neurons. So we're excited about this study. This is a researcher who's been quite productive and has really used those baby teeth that you all have sent to really begin to understand the very basic biology of how a PWS neuron is different than a typical neuron. So this is a unique resource that's being used, and it's a phenotype, or it's a characteristic. This delayed maturation is a characteristic that can potentially be used in drug screening and to assess the efficacy of a treatment in a dish. So a relatively easy way to look at new compounds and see if they might correct this difference in PWS neurons. So we're looking to this work to define targets for new therapies aimed at releasing this developmental block and restoring the PWS neurons to a more typical maturation profile. And complementing this work is work from a new investigator, Dr. Ladota, who is in Italy, and she has a lot of experience in some of the really cutting edge technologies and innovative systems using three-dimensional cortical structures. So essentially what we call brains in a dish. So taking the neurons from stem cells, or taking stem cells, changing them into neurons, and then allowing them to form into three-dimensional structures. And these three-dimensional structures are really important because they allow scientists to look at the cell-to-cell communication. So Dr. Ladota is an expert in those technologies, which are, again, really cutting edge, and she's been working with a lot of fantastic groups on that. She is new to the PWS field, but she is supported by some experienced PWS researchers. So it's a great combination of, experienced PWS researchers who can help her sort of understand the nuances of PWS with someone who has fantastic experience in using these technologies to really understand cell-to-cell communication and how brain function in an individual with a neurodevelopmental disorder differs from a typical neuron. And she's looking at cerebral or cortical neurons, which are important in, many of the thought, language, reasoning, and memory. So some of the cognitive aspects of PWS. So we're excited, again, because this is a new investigator who has experience in an area that is really, just moving along really nicely and helping people understand some of the changes in neurodevelopmental disorders. And it adds to our critical mass. We now have a number of scientists who are looking at different aspects of brain development, and we really want to bring those individuals together so they can share their findings and really help us tease out how brain development in the hypothalamus is different in PWS, influencing things like hunger and thirst, and then how brain development in the cortex is different, influencing things like cognition and memory. So in the long term, we hope this project will provide insight into how inactivation of the genes alter brain function. And also these organoids can be used, again, to be testing drugs to see if we can take a phenotype or characteristic that is PWS and use a therapeutic to have it behave in a more typical fashion. And finally, in the group of molecular studies, Dr. Rob Nichols is going to be looking at miniaturization of the PWS domain for a AAV-based genetic therapy. So AAV, adeno-associated virus, is a great gene therapy vector. My background is in gene therapy, so it's a really useful gene therapy vector. It tends to be very safe. It allows long-term expression. It can get up into the brain and deliver genes to the brain. The one limitation, or one of the limitations of AAV, is that it's actually it's a small virus, so you can't just sort of throw everything in the kitchen sink into it. So in order for it to be useful for PWS and for other disorders, researchers really have to do some sort of fine tuning and engineering of the genes that are going to be inserted and then potentially delivered. So Dr. Nichols is going to develop a novel single AAV that includes many PWS genes. So a lot of the PWS genes are small, and some of them can be sort of shrunk, and then they're going to be put together into this vector to see if that might be a way to replace the gene. So all individuals with PWS, we know, have inactive genes sitting there, and one way to address that therapeutically is to deliver active genes. And so this would be looking at delivery of active genes from the PWS region. So we're excited about this study because it is a novel gene therapy approach potentially to treating PWS. As we know, the PWS region is extremely complex, and Dr. Nichols probably knows it as well as anyone in the world because he's been studying it for many years now. So sort of that tinkering to get down to the core elements and be able to put them in an AAV is particularly well suited to his group. And we think that this would be a resource that might be useful for others. You could start looking at, how this vector, if it's delivered, for example, to a mouse or a rat model, would be able to restore function and, bring those animals towards a more typical development. So there's a lot of both in cell models and in animal models, these resources might be very useful. And Dr. Nichols is looking to use them in a beta cell model, so a model of pancreatic cells, and also in a mouse model of PWS. So I'll end with a few grants that we have funded that have more of a clinical focus. So again, this program, our grants program, funds things across the spectrum, everything from the really basic biology, which is so important to helping our therapeutics, all the way through the clinical side of things. And these are 3 projects that are important on the clinical side of things. So I'll just walk you through those. The first is a really important project, particularly in the context of the recent developments in the PWS field. So one of the challenges that we have in clinical trials in PWS is really showing that a drug is effective. And that has been a challenge for pretty much every drug that has come into the PWS field. And in some cases, we as parents, we can see that a drug is having an effect. And I'm thinking of LV-101, the levo drug in this case, which recently had that FDA advisory committee meeting that was unsuccessful. So we can see the differences, but the FDA has to be able to quantify that and see it and be convinced that there is a change in hunger in order to get the approval. And part of the issue that we currently have is the reporter-based systems, the questionnaires that we use. They're good. They capture many aspects of hyperphagia and PWS, but sometimes they're just not sensitive enough, likely to detect some changes that might otherwise be meaningful. So we need to build up that complement of endpoints and assessments that we can use in clinical trials. And so Dr. Gallagher is going to be building off some work that has been done at Vanderbilt University, kind of taking it to the next step, looking at eye tracking and attentional bias as a potential biomarker and endpoint in clinical trials. So this study is going to look at eye tracking as a biomarker for altered satiety in PWS using a computer-based system. This is a very experienced researcher. Her initial focus has been in autism, but she's also been doing some work recently in PWS, and she also has experience in developing endpoints for autism clinical trials. So she brings that experience to the study. And obviously, this is an area that we need to be bolstering up so that drugs that have an effect, but maybe not a meaningful, but not necessarily, dramatic across the board effect, can still demonstrate effectiveness in clinical trials so that we can reach approval. So the long-term goal is to create a robust, objective biomarker of hunger and satiety that can be used to monitor treatment effectiveness for hyperphagia in PWS. The next study is a bit of a different direction, but something that is really critical in our community. Anybody who has a child with PWS knows that it can be really taxing on the parents and or caregivers. And if you don't have a healthy parent, mentally healthy parent, the child will not thrive. And so Dr. Kelleher at Purdue University has been looking at ways to deliver support to families efficiently and in ways that can be scaled up and be effective in providing support for caregivers so that they can be better positioned to provide care to their children. And so this study is going to look at telehealth-based treatments for caregivers and determine what's most feasible, what's most effective. So it's really looking at, effectiveness of these different therapies delivered by telehealth to support caregivers and thereby support individuals with PWS. So we're excited about this study because This is a very enthusiastic research team that really is interested in supporting caregivers of those with neurodevelopmental disorders and has a lot of experience in that area. This is obviously an area of significant need in our community. Caring for an individual with PWS, we all know, can be quite challenging, and having that support to help us through, again, is really important for the health of the entire family. This will provide an opportunity for families to access support while at the same time advancing research. So this group will actually be administering these telehealth interventions and then measuring what works best for families. So we'll get the advantage of having those telehealth interventions and also, advancing the research, evidence-based research on what works best. If we have happy parents, we can have thriving child with PWS. So the long-term goal is to develop effective telehealth-based mental health services for the caregivers of individuals with PWS. And finally, the last project that we'll be funding in this round is a study by Dr. Ann Wheeler, who's at RTI International. And this is an extension of work done by Dr. Godler in Australia, developing a newborn screen. So we know that many babies with PWS are diagnosed in the newborn period, but there are still a good percent that slip through that initial diagnosis and may not get diagnosed for years. And that's a lost opportunity for those families to initiate growth hormone therapy, to learn about PWS and, start building a care team to take care of the individual with PWS. And so this study is looking at the feasibility of doing newborn screening. So newborn screen is that heel prick when babies are first born that, that every baby participates in across the country, and it screens for genetic disorders that can be treated and are severe and, would benefit from that early diagnosis. So FPWR has banded with several other organizations that also have chromosome 15 disorders. So the Angelman Syndrome Foundation, FAST, which is the Foundation for Angelman Syndrome Therapeutics, and the Duplication 15 Alliance, which is for individuals who have a duplication of the same region that we have a deletion of, which also presents problems. So Dr. Godler has developed a test which can detect all of those in one test, which makes it economically feasible to use as a newborn screen. But there needs to be additional work to show that it can actually be implemented on a large scale in the large scale population. So Dr. Wheeler and RTI will be incorporating that into their early check newborn screen platform. So this is a platform in North Carolina where families can opt in to have additional genetic disorders tested for in the newborn period. We're excited about this study. As I said, it's a collaboration among the chromosome 15 disorders. We all need each other. If we can do this test, as I said, in one economical test, then all individuals can be tested for all of those disorders, and we can all pick up those early diagnoses soon after birth. And, as I said, some babies with PWS are not diagnosed in that newborn period, so it would allow those individuals to be, detected and receive the early intervention care that they need. And, the other thing of interest is Dr. Godler's initial studies where he's looked at, I think 17 or 18,000 blood spots suggest that PWS may actually occur at a higher frequency, maybe 1 in 8 or 9,000 individuals, as opposed to the 1 in 15 to 30,000 that we often hear quoted. But we won't really know that until we actually test very large populations. So this will add to that information so we can get a real idea of the incidence of PWS in the general population. So this is a collaborative effort to establish the feasibility of adding a single test that would detect PWS, Angelman syndrome, and DUP15. And that would be the first step towards a nationwide newborn screen of those disorders. So that is the summary of our newly funded grants. What can we expect as we wrap up 2021 and head into 2022? As I mentioned, we have a lot of clinical trials that are either open or will be open very soon. So please, if you can, sign up for our clinical trial alerts so you can learn about those and see whether you would be able to participate. I don't know how many of you participated in the conversation we had, FPWR and PWSA, shortly after the advisory committee meeting for LEVO. But I think Elizabeth Roof was particularly eloquent when she said, you know, in order to get these clinical trials done, we need butts and seats. So, you know, we are the only ones that are going to be able to get these clinical trials done. So please consider participating if you can. We do look forward to the next step on the recently completed phase 3 trials. LEVO is considering options and will let the PWS community know as soon as it gets feedback from the FDA and decides on next steps. Soleno, again, is looking at or having discussions with the FDA about whether it is time to submit a new drug application. So of course, we'll be keeping a close eye on that as well and are very excited about that possibility. We'll be looking for new insights into PWS biology and molecular mechanisms based on the previously funded studies, of which we're getting some really interesting new insights into PWS and the continued development of new targets for treatment of PWS. We're also looking in 2022 to really make a push to make sure that we're disseminating the results out to the community so that you can use the information that our funded projects are generating. An example of that is the social skills. So we've funded a couple of projects on improving social skills, both in young people with PWS as well as teens and adults. And we're working with those researchers to develop resources that you can take maybe to your teachers or to your early intervention team to assist in sort of those PWS specific aspects of social growth that need to happen. And finally, we'll be looking for the development of new projects from the PWS clinical collaborative, and please look for those and participate in those as you can. So that's all I have. Susan, if you want to look at the questions.
Absolutely. Thank you so much, Theresa. And just as a reminder, if you had any questions from today's presentation, you can just place those into the Q&A chat box and we will answer as many questions as we have time. We have a few minutes here that we can do that. Theresa, a couple questions came in. First, let's start with the newborn screen. You mentioned that it's starting in North Carolina. What needs to happen in North Carolina in order to bring that test to to some other states?
So the newborn screening system in the US is, it's really complex, I guess is the easiest way to say it. So first we have to establish that it's feasible. So the goal is with the North, so it's been done to some extent in Australia, and the goal is to bring it to the US and show that in North Carolina, A, that they can get the technology to work reliably, and then B, that it's economically feasible to do. You can't have a test that costs, you know, $100 per child if you're going to screen every newborn. It has to get down to, you know, less than $5 per child. So once they establish those things, then there's another component that we are working with clinicians, and there's several clinicians in the PWS field who are really interested in this. Virginia Camonis is one. Establishing the rationale. You have to be able to demonstrate that it is important to get that early diagnosis, that it will make a difference. And for PWS, you know, we will argue that getting growth hormone during infancy is really critical for brain development and, you know, and getting that education early. For the other disorders like Angelman syndrome, they're actually developing some gene therapies that are likely to start in the next few years. And those gene therapies may be best suited for newborn babies. And so that's the argument that they'll use. And we kind of all go together. So we're sort of bonding together and trying to make the rationale, because you have to convince, there's what's called the RUSP. You have to make a case that it makes sense to screen for the disorders. So it will likely be a long path. We'll have to show that it's feasible in North Carolina. We'll have to build the clinical case that the screening should be done. And then we'll have to bring an application to the RUSP, and they'll review that and decide whether to recommend it. And then it goes state by state. Each state health department has to be willing to adopt that. So it's a very long, complicated process. So it will take a while, but we think it's really important to make sure that every baby with PWS is diagnosed in the newborn period. We suspect that, there are underserved communities, be it, out in rural hospitals, potentially different ethnicities that, aren't sort of may be underdiagnosed because it's not what the textbook looks like, for example. And if we had an unbiased screen that was available across the country, we would make sure that we were capturing everybody who needs to be captured.
Sure. So people with Chauffe-Yang syndrome only have a single disrupted gene, which is MAGEL2. Will this test, is it sensitive enough to identify people with Chauffe-Yang syndrome as well?
This test would not. So this is a methylation-based test, which would not detect Chauffe-Yang syndrome. There are other tests that are being developed, even like whole genome sequencing, that is that some states are trying to implement for babies that show up in the NICU, for example. So there may be other approaches that are particularly for those babies that have complications right at birth, that may be available in coming years.
Mm-hmm. You mentioned Dr. Ryder's work and that many of us have submitted our baby teeth to him. Do you know if he's still accepting baby teeth for future studies?
I believe he is. His contact information, I think it's L. Ryder at it's University of Tennessee Health Sciences Center. So it's Larry Ryder, University of Tennessee Health Sciences Center. He has a web page, and I think his contact information is right there. And if you can't find it, shoot me an email and I can send it to you. But you do have to have the kit ahead of time, because once the tooth falls out, it has to get popped right into a tube with media in it. So it's if you would like to donate, please contact the Ryder Lab and they'll set you up. I think they are still accepting teeth.
Mm-hmm. So now he has used those teeth to make neurons. And you mentioned that we're now going to start screening drugs to see if we can help fix some of the challenges that he's identified. How are these compounds going to be found or selected?
Well, he's initially using compounds that are, a few compounds that have been suggested that might be useful in PWS. So he's just got to sort of, by reviewing the literature, he's got a handful of compounds that way. You could also envision, and one thing that I think it's time to get our cell model experts together and discuss, you could envision a high throughput assay. So there are these libraries with thousands of compounds in them. Once we have a phenotype that is really robust and can be quantified in an automated fashion, we can think about doing a high throughput screen with an unbiased high throughput screen. So where you just throw thousands of compounds on the cell and then see if the cell changes in a way that you think would be associated with an improvement of conditions. So I think that in the initial go around, he's going to be testing the robustness of the phenotype and then using compounds, that we know may have an impact. And that will set us up to do an unbiased screen with thousands of compounds.
Well, that's great. It sounds really exciting. I look forward to hearing more about his work. We did promise a 45-minute webinar. We're now at 50. So I would like to thank you, Theresa, for presenting this update. It truly was great to hear all the work that's being done. And a huge thank you to everyone who joined us today. This webinar has been recorded and we will share it on our YouTube channel, along with our other recorded webinars and conference presentations. So if you haven't already, please check out our channel. We did include the link in the chat. However, if you've missed that, you can always go to our website at fpwr.org and click on the YouTube icon, which is found on the left sidebar. So thank you for joining us and have a wonderful rest of your day.
Thank you.